Good morning, and thank you for the invitation to appear here in this bustling city that is having so many questions, but also quite some answers to biotechnology. So what I'd like to do, how do I shift here for the next slide? You do that? Okay. Yeah. What I'd like to guide you through is perhaps a little bit of a different aspect than Pat has so nicely introduced, and that is more the manufacturing and its physical presence of this. The manufacturing is, so it is more to do with the physical manifestation of the manufacturing facility and what needs to be housed in it and what impact it has on the manufactured good. So I'd like to go, before we go into the future, see a little more about where we come from, what has it been so that we reinvent mammalian cells to be useful, and what do we mean by capacity, because capacity is, I think, always been tossed around it, and the industry had constantly been either under capacity or over capacity, but never at capacity. So let's see what this means. And from there, the design of plants as they have evolved and where they may go in the future. So here is the short, only a very short and personal history of the very short-lived but off-age biotechnology. I think the founding events were the discoveries of the plasmid, the Cohen-Boyer, and the Cohen-Milstein discovery of the monoclonals, all in the early 70s. And so this was also the time when some of the now eldest companies in this field were founded. To come in, to market the insulin was the very first. At that time it was an E. coli product, and the agency, there are some similarities to today, what to do with biosimilars, wasn't quite capable of really deciding how that should be done. And so the approval process for the insulin at that time was still very much like a small molecule. Then it came later, not very far away. It was a mammalian cell product, tissue plasminogen activator for the dissolution of blood clots. That actually also generated a major stir in the manufacturing technology because it was a mammalian cell and it needed, because of low titers, a lot of volume. Then another one that came from the monoclonal was the Muramab, or OKT3, if people still remember that. The manufacturing didn't seem a problem then, but I don't think that it would be approved today because this was milking ascites mice with the antibody, and it was a murine antibody. So this was the early experience with manufacturing of recombinant proteins by cells, be it microbial or mammalian. The big breakthrough, money-wise, was somehow with EPO, and then later also with Avanex, or in particular with Enbrel. And so this went well into the 2000s, and we have there then also little experiences that mitigates our euphoria about mammalian cells or biotechnology, namely that, for example, from a manufacturing point of view, the lack of not being able to supply actually was the reason why Immunex couldn't make as much out of it and was actually bought over by Amgen. And the other time and reason for thought was an experience that we know everything about antibody, we thought, and then therefore a phase 1 clinical trial is easy to do. Not so. In the De Janeiro event, there was an antibody trial in which the first patient, one patient died and 6 patients were seriously ill, which stimulated even more in the agency, the European as well as the FDA, to look into safety aspects. And as Pat already has mentioned, these safety aspects are not very obvious to be found, since the molecules that we deal with are actually bypassing our immune defense and going into systems directly, and what they do there often enough is perhaps not very predictable from in vitro systems. To see that nonetheless this has been quite a successful industry, you can see that, and you don't need to go through all these lines, but you can see that their line, this is the top 10 sales of monoclonal antibodies that in 2008 had about 30 billion in sales, and what is somehow indicative that most of these are antibodies or FIV fragments, but the one that actually scoops all the antibody is actually a hybrid, and that is entanercept with 8 billion. It's still the largest single manufacturing antibody-like product in the world. Then followed by Remicade and Rituxan, but these are of course very interesting competitions, and we will see where this will land in the future years. Now with Rituxan having even more biological activities than thought. So, the evolution of the production methods of these very valuable pharmaceutical proteins and the dependency on quality, I just want to go there because quality is one of the major things that has an impact on it. And so here is another one. This is the same timeline, only now from the discovery of the monoclonals comes what type of facilities have been built. In the early days, this was not much of a single event. The facilities were taken from microbial for E. coli, but for mammalian cells, they needed to be scaled up, and the first 10,000 liter bioreactor was developed and put into use by Genentech and Boehringer Ingelheim at the time when TPA seemed to be a very interesting perspective, an enzyme that could be made to dissolve clots in myocardial infarction. Rituxan, the first antibody, mentioned the antibody itself was this hybridoma was 75. It took more than 20 years to bring the first therapeutic monoclonal antibody to market. It's quite remarkable. So the evolution that took place over all that time was an evolution that started with cells as a substrate. So when we talk about biosynthesis, then actually what all we do is we allow cells to do the synthesis, but we see to it that the cells are so well cared of that they can do that well. So this is a very indirect synthesis, very different to small molecules where we do the synthesis itself. And as from the vaccines area, they were primarily in the mammalian cells, only adherent cells, and this is when most cells in the bodies are adherent and very few are suspended, but there's a persistent technical problem to get in scale to get a lot of these cells. So the breakthrough came in somewhat of a competition that I think combined efforts of Genentech and Boehringer Ingelheim trying in a joint venture to get TPA cells. And it was really, on a personal note, a competition between can we get enough adherent cells to make it, or can we convince the cells to grow in suspension? And if we can grow them in suspension, then we can use deep tank technology. We could achieve both, but for the future it was better to do them into suspension. And so it came that the persistent cells learned to swim and were immobilized and were adapted to grow without cell adhesion, and this produced what is still the workhorse as a host cell in the biotech industry for mammalian cells, Chinese hamster ovary cells. Not really destined for making antibody or anything, but it is interesting to know that the origin of these Chinese hamster ovary cells actually dates back to 1905 when these cells were taken into culture in attempts for monitoring some immune reactions. So these then also put into perfusion cells. Perfusion in cartridges, hollow fibers, are still sometimes used for diagnostic antibodies, but not so much for therapeutic or in man usable. And the expansion came into deep tank, and this is what is today our standard platform. So what we have is, we have a gene on the plasmid that is put into a cell, that is put into a master cell, and it goes through all these expansion, harvest, and purification to eventually come as something that is in a vial or a syringe. And this is the normal process that is also a standard process. Cell expansion, separation of the cells from the product, and the downstream to purify, and finally to put this into something that the patient can use or how it is being applied. This is another schematic look at it from the very early time. The cell bank are very few cells comparatively, in millions, and they are grown. These cells all, you know, they do not like to be by themselves, but they do not like to be too many either, so you have to have a window, and with increasing cell mass that you need in order to make your recombinant, you need more volume. At the largest volume, you then separate the cells during all that time, secrete the antibody or the protein in question, and in the supernatant is collected, and the cells are being taken out as particles by various means, and this is the feedstock then from a purification that usually consists of 3 columns and a number of ultrafiltration or filtration steps to actually end up in something that is the so-called bulk formulated product, drug substance.